Why do introns exist? A curious genetics student who first learns about the concept of introns and RNA splicing often wonders that why introns exist. If intron sequences are destined for removal, then why are they there in the first place? Wouldn't it be more efficient if introns were absent and hence never transcribed? Indeed, scientists asked these same questions shortly after introns were discovered in 1977. However, we know now that introns indeed serve several functions. But let's discuss the basic concept about the introns first. Remember that DNA is a sequence of nucleotides. The simplest form of a gene is a length of DNA. RNA is sequence of nucleotide. Polypeptide is sequence of amino acid. DNA has exons and introns. Then there is RNA synthesis. Interrupted genes are expressed via a precursor RNA, having both exons and introns. Introns are removed when the exons are spliced together. The mature messenger, RNA, has only the sequences of the exons. Introns are the intervening sequences that are removed when the primary RNA transcript is processed to give the mature RNA product. Then protein synthesis occurs. Now, let's discuss the key points in it. The processing of interrupted genes requires an additional step. That is not necessary in uninterrupted genes. The DNA of an interrupted gene has both exons and introns. The DNA of an interrupted gene is transcribed to an RNA copy, called as a transcript. That is exactly complementary to the original DNA sequence. This step is called as transcription. This RNA is only a precursor, though it cannot yet be used to produce a polypeptide. First, the introns must be removed from the RNA to give a messenger RNA that consists only of a series of exons. This process is called RNA splicing and involves precisely deleting the introns from the primary transcript and then joining the ends of the RNA on either side of each intron to form a covalently intact molecule. During splicing, the exons are always joined together. In the same order they are found in the original DNA. So, the correspondence between the gene and polypeptide sequences is maintained. Then there is protein synthesis, called as translation. In eukaryotes, transcription occurs in the nucleus, and translation occurs in the cytoplasm. Let's memorize it using a mnemonic. Exons are expressed and introns go in the trash. Moving forward, we might envision the simplest possible mechanism for removing an intron to involve two steps. First step is that the intron is cut at both ends by an endonuclease, and the second step is that the adjacent exons are joined, or ligated, by a ligase. This is, apparently, what happens to the introns. present in transfer RNA in bacteria. However, in studies of other RNA, such as transfer RNA in higher eukaryotes, and ribosomal RNA and pre-messenger RNA in all eukaryotes, precise excision of introns is much more complex and a much more interesting story. Moving forward into further details, introns in eukaryotes can be categorized into several groups based on their splicing mechanisms, such as Group 1 introns and Group 2 introns. We will discuss them one by one in detail. Group 1 introns, such as those in the primary transcript of ribosomal RNA, require no outside help for intron excision. The intron itself is the source of the enzymatic activity necessary for splicing, called as self-enzymic activity. This amazing discovery was made in 1982 by Thomas Sech and colleagues during a study of the Ciliate protozoan Tetrahymena RNA that are capable of such catalytic activity are referred to as Ribozymes. Ribo means RNA, and zyme means enzyme. Self-excision of Group 1 introns is known to occur in preliminary transcripts for messenger RNA, transfer RNA, and ribosomal RNA in bacteria, lower eukaryotes, and higher plants. Now, moving into further details, and let's discuss the self-excision process for Group 1 introns. Chemically, two reactions take place. These are exons and intron. Other is hydroxyl group and guanosine. While this is a active site within intron, in next step, interaction between a free guanosine, symbolized as G, which acts as a cofactor in the reaction, and the primary transcript. Then, in next step, after guanosine is positioned in the active site of the intron, its hydroxyl group attacks the phosphodiester bond, presented as P, in next step. After attacking its hydroxyl group, breaks the phosphodiester bond. The second reaction involves the interaction of the newly formed hydroxyl group on the left-hand exon and the phosphodiester bond at the right intron exon boundary. Then, in final step, the intron is spliced out, and the two exons are ligated, leading to the mature RNA moving forward. As we have discussed that, there are several groups of introns, such as Group 1 introns and Group 2 introns, and we have discussed about the Group 1 intros. Now, let's discuss Group 2 introns. Splicing of Group 2 introns is somewhat different than for Group 1, but also involves two autocatalytic reactions, leading to the excision of introns. Autocatalytic reaction is the reaction where a product itself acts as a catalyst for the reaction. It can act as a catalyst for the same reaction, or the coupled reaction. It increases the rate of the reaction. Group 2 introns are found in fungi, plants, protists, and bacteria. After understanding the basics, Now let's discuss. Why do introns exist? Well, we know that. Introns indeed serve several functions. That's why introns exist. So, let's discuss functions that require existences of introns in details. Some genes can encode for more than one protein product through the alternative use of exons. This process, known as alternative splicing. Introns may also be important to the evolution of genes. Once an intron is excised from a pre-messenger, RNA, it is generally degraded. However, there are many documented cases where an intron actually contains a functional non-coding RNA. In such cases, the excised intron is processed to liberate the non-coding RNA, which then functions within the cell. Non-coding RNA function to regulate gene expression at the transcriptional and post-transcriptional level. Introns can also regulate transcription. For example, intronic sequences in the DNA frequently harbour cis-regulatory elements, such as enhancers and silencers, that upregulate or downregulate transcription, respectively. Let's discuss them one by one in detail. First is alternative splicing. Let's understand it. Some genes can encode for more than one protein product through the alternative use of exons. This process, known as alternative splicing, produces different mature RNA from the same pre-messenger RNA by splicing out introns and ligating together different combinations of exons. This means that a eukaryotic genome can encode a greater number of proteins than it has protein-coding genes. Second function of introns is evolution of genes. Let's understand it. Introns may also be important to the evolution of genes. On evolutionary timescales, DNA sequences may be moved around within the genome. The modular exon/intron gene structure allows for new genes to evolve when, for example, an exon is introduced into an existing gene. Third function of introns is liberation of non-coding RNA. Let's understand it. Once an intron is excised from a pre-messenger RNA, it is generally degraded. However, there are many documented cases where an intron actually contains a functional non-coding RNA. In such cases, the excised intron is processed to liberate the non-coding RNA, which then functions within the cell. Non-coding RNA function to regulate gene expression at the transcriptional and post-transcriptional level. Fourth function of introns is that introns can also regulate transcription because intronic sequences in their DNA frequently harbour cis-regulatory elements, such as enhancers and silencers. Let's understand it. Introns can also regulate transcription. For example, intronic sequences in the DNA frequently harbour cis-regulatory elements, such as enhancers and silencers, that upregulate or downregulate transcription, respectively. Cis-regulatory sequence that increases the activity of a gene, when bound by transcription factors, is called an enhancer, while a sequence that causes a decrease in gene activity is called a silencer. So, this was all. Thank you for learning. Join us again.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.